Hello students, welcome to Capman Academy. So today we are going to discuss about the carbohydrates. We are going to continue our previous class. So in the last class we had discussed about the general properties of the carbohydrate, right? We had seen what are their functions, what are their definitions and what are their classification. So in the classification, we had seen that your carbohydrates are actually categorized into different categories according to their structure: mono, di, oligo, and polysaccharides. And also we had discussed about the features of monosaccharides, how they are being synthesized, what type of processes are actually involved in the carbohydrates, right? So in our today's lecture, we are going to discuss about rest of the carbohydrate categories, for example, disaccharides, oligosaccharides, and our polysaccharides, right? So in the disaccharide cases, as the name speaks, it has two sugar molecules which are actually being joined together. If we are talking about the glucose, then glucose will join together to another type of glucose molecule and it will form disaccharide. So when there are two sugar molecules in the structure, that type of sugar is called disaccharides, right? And if they are more than 2 or up to 10, then these type of sugars are called oligosaccharides. So disaccharides are also called as oligosaccharides sometimes, right? So now the question comes, how they actually being joined together? What type of bond is actually responsible here, right? And what are the feature of these structures? So we are going to taking the examples of the disaccharides, the famous one, for example, the sucrose, lactose, and the maltose, and going to see what different type of features the disaccharides have, right? So first of all, you need to remember they actually differ in the sugar moieties. They can differ in the sugar moieties. Second, you need to remember their bond, which is actually joining them. And then third, you need to remember whether your sugar is going to be reducing sugar or it is going to be non-reducing sugar. So these are the very important aspect of your disaccharides. So we are going to discuss one by one about these disaccharides, right? So if we are talking about lactose, let's discuss first about the lactose. So in case of lactose, two sugar molecules will join together. And what are these sugar molecules? Number one is your galactose and second one is your glucose sugar molecule, right? So first answer here is that lactose is composed of galactose and your glucose sugar molecule. This is the first point, two sugar moieties. Second point is what type of linkage they have. So this linkage, when two sugar molecules they join together, this type of linkage is called glycosidic linkage. Glycosidic linkage, right? Now you need to remember at what position this glycosidic linkage is being formed. So in case of the lactose, this glycosidic linkage is called beta 1,4 glycosidic linkage. Sorry, it is being denoted as like this one. Beta 1,4 glycosidic linkage. How? If you see in the structure, your first sugar molecule here, it is in the beta anomeric form, right? And it is joining or making the bond with the next carbon, next sugar, which is next sugar at the position of fourth carbon. So this one is the anomeric one, beta 1, and the fourth carbon position of the second sugar unit, that one is called beta 1,4 linkage. So this bond is being formed between the two sugar molecules beta 1 and 4 position. So this is the second answer. Next thing comes whether your sugar is reducing or non-reducing sugar. Reducing means whether this lactose can actually react with other type of molecules, other sugar molecules, you can say, right? So if we see its structure, its alpha-meric carbon in this sugar molecule is free, right? Because here the fourth carbon is bonded with the sugar molecule. So first anomeric carbon here, it is free to bond with other sugar molecules. So this type of situation is called reducing sugars, right? This situation is called reducing, when your sugar can react with the other sugars, right? So these type of molecules are called reducing sugar. So lactose, if you say, your lactose is a reducing sugar molecule, right? Now next come that what type of the enzymes actually hydrolyze the lactose. So lactose hydrolyzing enzyme is called beta galactosidase or sometime lactase. ठीक है? So this enzyme actually it hydrolyzes this linkage, beta 1,4 linkage, right? And it actually breaks them into the galactose and into the glucose sugar molecule. So in our body, how the lactose is actually being digested? In that case, in our gut we have beta galactosidase enzyme, right? And this beta galactosidase enzyme, it actually breaks the lactose into the galactose and into glucose, right? And after that, the both components are being used for the metabolic activities in the organisms, right? So when there is the absence of the beta galactosidase in the gut, these type of situations are called lactose intolerance, which means your body cannot digest the lactose, and lactose is present in the dairy product. So milk product, you must have seen that some products, some people, they cannot digest the dairy products, right? They got some type of allergy. So that is due to, in some cases, they have the lactose intolerance in which they lack the enzyme beta galactosidase enzyme, which is not able to digest the galactose and glucose, and in the result it actually do the fermentation and causes the pain in the stomach, gas and bloating in the stomach, right? So sometimes it occurs due to the beta galactosidase enzyme activity. So these type of people who cannot digest the dairy products, they actually have the lactose intolerance, right? So this is about the lactose. First, it is made up of galactose and glucose sugar moieties. It has the beta 1,4 linkage, and its one anomeric carbon is free to react with other sugars. So this is a reducing sugar molecule, right? And it is being hydrolyzed by the enzyme which are called beta galactosidase, in which in the absence of the beta galactosidase, the conditions comes which is called lactose intolerance, right? So our next sugar, that one is called maltose sugar. Maltose sugar is also your disaccharide, and it is consist of glucose moieties, right? Glucose 1 and glucose 2, right? And it has again the O-glycosidic linkage. So first question answer here, this is consist of glucose and glucose. Second, O-glycosidic linkage. Now its first carbon molecule, anomeric carbon, is free here, and 1, 2, 3, 4 and 1, and here the position is the alpha anomeric. So this bond is called alpha 1 4 linkage. So the glycosidic linkage in your maltose sugar is alpha 1 4 linkage, and it is occurring in between the two glucose molecules. Now it has a free anomeric carbon, so this one also your reducing sugar, right? These type of sugars, they are also called malt sugars, right? Why? Because when your complex homopolysaccharides, they actually breaks into the simple sugars, they actually make the maltose sugars. For example, if we are saying, if you are breaking the longer glucose chain by using the degrading enzymes maltase, in that case you will get two pair of glucose which are actually your maltose sugars, right? So malt sugars are actually also being produced from the complex homopolysaccharides. We will discuss about them later. So this you should know. So this is also the malt sugar, they are also the end product of your starch. Sometimes question come, the end product of the starch molecule is what? So the end product is malt sugar, right? Our next disaccharide, that one is the sucrose, which is called table sugar also, and this one consists of glucose and fructose sugar moieties. But in the sucrose case, your glycosidic linkage changes. It is different than others. If you see in the structure of the sucrose, your fructose sugar, it actually flips its structure, right? As we are seeing here, this is the straight structure, and when there is a going to synthesize sucrose, in that case fructose actually it flips its structure to left side, right? So in this case what happens, the first alpha anomeric carbon, it has to get engaged with the linkage, right? So if you see in this case, your fructose is actually involved in the bond making, which means that your alpha sugar here, this sugar is alpha at first position, and it is making the bond between the fructose at 2 carbon position. So this one is called alpha 1 2, right? So linkage for the sucrose here is alpha 1 2 glycosidic linkage. It is made up of glucose and fructose, and now there is no reducing end here present in the structure. So this sugar is called non-reducing sugar, which means sucrose cannot react with other sugar molecules, right? So sometimes question can be asked, which one of the sugar molecule is the non-reducing sugar, which one of the molecule is the reducing sugar? So all the disaccharides in that, only the sucrose is the non-reducing sugars. How? Because its free anomeric carbon is actually engaged with the glycosidic linkage, right? So sometimes because the sucrose, it is actually a table sugar, some people they do not or cannot actually consume it due to some conditions, right? Some health conditions. So in that case, you can also synthesize different type of the artificial sweeteners. In those artificial sweeteners, actually they will taste like sweet, but actually they will not have any type of the calories. And these artificial sweeteners are called your acesulfame, your aspartame, and your sucralose, etc. Okay? There is another one which is called stevia. This is also artificial sweetener, but this is being extracted from the plants, and that plant is your stevia, right? So these actually sugars are called artificially synthesized sugars, and they actually does not have any calories, and they are actually preferred for those people who actually suffers with the diabetes or with some other conditions, right? Next topic is the polysaccharides. So with this, we had discussed about all the disaccharides, important disaccharides, sucrose, lactose and maltose. So you should know about their sugar moieties, the bonding and their feature reducing and non-reducing sugars. Okay, we haven't discussed about the enzyme. So the sucrose is actually being hydrolyzed by the enzyme which is called invertase. Right, so invertase actually it cleaves the bond between glucose and in fructose and leads to the synthesis of glucose and fructose. So for the sucrose, invertase enzyme is being used for the hydrolysis, and this one is also called invert sugar. Invert sugar, because in this structure your fructose is actually changes its structure, it gets flipped, or you can say invert. Right, so next let's discuss about the same way your oligosaccharides are also being formed. They could be, there could be tri sugar molecules, 3 sugar molecules, there could be 4 sugar molecules. You should know about all the examples. Right, now let's discuss about the polysaccharides. So polysaccharides are actually very complex structures. They are very complex structure and they have the sugar molecules of more than 1000 units. Right, so let's discuss about them. So your polysaccharides are actually basically categorized into two type of categories. Number one is called homopolysaccharides and second one is called heteropolysaccharides. Right, or they are called glycosamine glycans or GAGs. Right, so we will discuss about one by one about them. So your homopolysaccharide means in this case your sugar molecules will be same. If there is a glucose, then in all the chain there will be glucose sugar. So these are called homopolymer. In this case the sugar moieties will be different in the chain. Right, so your homopolysaccharides, according to their structure and bonding, they are actually being categorized into two categories. Number one is called storage and second one is called structure, which means the saccharides which are responsible for the storage of the glucose or sugar molecules, those are called storage, and which one actually involved in the structural stability of your cells, those are called structural polysaccharides. Right, and the examples, in case of plant there is starch, in case of animal there is glycogen. Structure, in plant there is cellulose, in the animal there is chitin. So let's discuss about these two type of the homopolysaccharides, storage and structure. So this is, I think, clear. What are the storage one, what are the structural one? Right, so first of all your starch, which is a homopolymer. Now in this case you also need to remember number one, their features, which are whether they are homopolymer or whether they are heteropolymer. So we are discussing about starch here. So first of all, this is homopolymer, which means it has single glucose moiety in its structure. So it will repeat itself again and again. Right, and what type of the sugar molecule? In starch there is glucose, or you can say the D-glucose sugar unit will get repeat one by one. Right, now in the starch there are two type of subunits are present. Subunits of homopolymers are present. Right, and these subunits are number one, amylose, and second one is called amylopectin. Right, amylose, this structure is actually will form only the linear chain structure, whereas amylopectin it will form the branched structures. Right, so linear chain, it also have a bonding in between. For example, this is a linear chain of glucose, then this bonding is called alpha 1,4 bonding. You need to remember the linear amylo structure, it has alpha 1,4 linkage, whereas your amylopectin, when it is in the linear, form, it has alpha 1,4 structure, 1,4 linkage, and when it is making the branched bonding, then it is having this one bond is called alpha 1,6 structure. So this is the difference between amylose and amylopectin, right? In the linear chain of amylose, it has alpha 1,4 linkage, and amylopectin has alpha 1,4 plus alpha 1,6. Sometime question can be asked, in the case of branching, what type of linkage is present in the starch? So that is your alpha 1,6 linkage. Right? Now the question comes, this branching, how it will get decided, that how this branching will start. So in case of the starch, after 25 to 30 sugar molecules, your branching will get initiated. Right? So after 25 to 30 sugars, your branching structure, या फिर branch formation will start to get initiated. Right? Now let's discuss about the enzymes which actually degrades the amylose and amylopectin. So as we see that amylose is actually present in the saliva. So when we actually consume the starch, it starts to get digested in our mouth by the enzyme amylase. Now amylase is of two types, alpha amylase and beta amylase. Right? So they actually degrades the linear structure of the amylose. Right? Then next come the degradation of the amylopectin. So in that case, amylopectin is being degraded by the enzyme which is called alpha glucosidase. Right? Or you can say the debranching enzyme. Second one is your debranching enzyme. So this debranching enzyme actually targets both alpha 1,4 and alpha 1,6 linkage. Right? So that's all about the starch. Now let's discuss about the another. So I have a picture here. We can discuss here also. So as we discussed in case of the starch, it is present in number one. It is present in the plant. Very, very important point. It is present in two forms, amylose and amylopectin, linearized form and in the branched form. The linkage here, the bond are your alpha 1,4, 1,4, 1,6 bonds in the amylopectin. Branches, amylose does not have any branch, whereas amylopectin, it has branch, and after 20 subunits, your branching gets initiated. So if you see the structure of the amylose will be like this, and structure of the amylopectin will be like this. When they become more complex and get into a shape, they actually looks like this one. Right? So this is the starch structure and its features. Now let's talk about the another homopolysaccharide. That one is called glycogen. And glycogen, as we know, it is present in animals as well as in the human beings. We are also included in the glycogen. Right? So human being, they also contain glycogen as a storage molecules. Right? Storage means that we actually store the extra energy molecules in our body. How? As I told you that you consume the starch, it gets degraded again into the simplest sugar units. So simplest sugar units will become glucose, and they will form from the glucose carbon chain, like this one. So now in your intestine, now you have the digested starch, which is actually present in the sugar molecules, this simple sugar molecules, right? So in our body, what it does, it actually absorb these sugar molecules, right, and use them for different activities, for metabolic activities and for different type of biological activity. And the excess glucose, which is actually still present in the intestine, that is actually being absorbed and being stored in the body for the emergency purposes, right? So this stored one is called your glycogen, and this glycogen is actually being stored in your liver and muscle cells. So कई बार क्वेश्चन पूछ लिया जाता है that glucose is actually being stored in liver and in your muscle cells. ठीक है? And this one also, it contains amylose and amylopectin form. Same features and same bonding, alpha 1,6, alpha 1,4. But in this case, in case of glycogen, your branching, it starts after 8 to 10 sugar units. In case of the starch, it is after 20 sugars. In case of the glycogen, it is after 8 to 10 sugar units. Right? And when you actually need this energy, then you have to again degrade the glycogen into the simple glucose molecules, and that is done by the enzyme. Those are called glycogen phosphorylase, glycogen phosphorylase, and your glycogen debranching enzyme, glycogen debranching enzyme, right? So this is about the glycogen. So let's see what is left in the image. So glycogen, as we understood, that it is present in the animals and it is stored where? In your liver and in your muscle cells. And the simplest sugar is your alpha glucose, and it has alpha 1,4,1,6 amylopectin form, and it has branching, and it has branching after 10 subunits. So structure would be like this, and it will become like this in the compact structure. So in your glycogen or in your muscle cells, you have multiple glucose units stored in them. So when you actually, in case of emergency you need, then this type of glycogen storage is being hydrolyzed and energy is being released into the blood system, right? So this is about the glycogen. Next, homopolysaccharide, that one is your structural one, and that one is called cellulose homopolysaccharide. So this is also your homopolysaccharide, right? Second, it has variety in its linkage. Variety means it is different in the linkage than your starch and glycogen. It has beta 1,4 linkage, right? And it has sugar units glucose, right? So due to this beta 1,4 linkage, now your cellulose is actually make these like of linear sheets like structure, right? It does not make any branch structure, and due to this structure, they are called, due to this bonding, they are called structural homopolysaccharides, right? And they are actually important for the maintenance of the shape and integrity of your simple cells, right? Now, in humans, we do not have enzyme which degrades the cellulose, which is your cellulase enzyme. So sometimes question can be asked, humans, they do not have cellulase enzyme. Therefore, humans cannot digest the cellulose which is being digested, which is being consumed from the plants, right? Next polysaccharide is our chitin. chitin. So this is also your homopolysaccharide, right? And it has different linkage, which is called your N-acetyl D-glucosamine. D-glucosamine. So this is the single sugar unit which actually makes the polysaccharide structure, and it has a linkage again beta 1,4. So due to this linkage, they are also participates in the structural function, right? And it is present in insects, for example in the outer covering of the cockroaches or the beetles. Okay, so their covering is actually have the chitin on them. So these are the homopolymers. Now let's talk about another type of the polysaccharides, and those are called heteropolysaccharides. So I have, okay, in the image I also have the cellulose one. Source is plant, right? It is present in the plant. In plant where? In their cell wall structure. Subunit beta glucose. This also need to remember that cellulose, beta glucose, alpha glucose and alpha glucose. Bonds 1,4. Branches, no, there is no branching, right? And it is aligned as like this one, and they have the sheet like structure. Okay, so these are the shape of the cellulose, and same shape would be for the chitin polysaccharide also, right? So now let's talk about the heteropolysaccharides. So heteropolysaccharides, as I told you, they are also known as GAGs or glycosaminoglycan, right? And they are actually, as we say, this is your cell, then they are present in the extracellular ECM, extracellular matrix between the cells, right? They have some important features. Number one, their feature is they are highly negatively charged molecules, right? And second feature is that they are composed of repeating disaccharide. So this you need to remember. In this case, repetition of single monosaccharide sugar is not happening. The disaccharide sugar molecule is being repeated here, right? So it is a disaccharide repetition. So what type of disaccharides are being repeating here? So these disaccharides are your monosaccharide derivatives, which we had discussed in the previous lecture. And these monosaccharide derivatives are your acidic sugars and your amino sugars. So they both will make a disaccharide, and this disaccharide will actually get repeated again and again, right? So I told you that this GAGs, they are actually present in the extracellular matrix. So which means you have a cell membrane, and here they will be present like this one structures, right? So here you will have acidic and amino sugars. Now they have to get attached here in the cell membrane. So your heteropolysaccharides, they actually attaches with the cell membrane proteins. So now there is another structures being involved. So there are proteins present in the cell membrane, and these proteins makes the bond with a linker sugar molecule, that is your xylose, then galactose, then your glucuronic acid, right? And then there comes your disaccharide, repeated disaccharide units A and B. So this is how your heteropolysaccharides are actually being attached with the cell membrane. They are attached with the protein with a glycosidic bond. Glycosidic bond. Right? And with the small oligosaccharide linkage. Right? So this type of structure, when there are proteins and sugar molecules are involved, they are called proteoglycans. Proteoglycans. Right? So let's talk about this one now. Now I'm going to discuss about only this disaccharide repeating units. What are these units and how many types are there? Right? So these units are actually based on their structure and based on the negative charge on their structure, they are categorized into different forms. So number one is called chondroitin sulfate. Number one is your chondroitin sulfate. Second one is called your keratin sulfate. Keratin sulfate. Third one is called heparin. Fourth one is called heparin. Heparin. Fifth one is called dermatan sulfate. Dermatan sulfate. And sixth one is called hyaluronic acid. Hyaluronic acid, or this one is also known as hyaluronate. Hyaluronate. Okay, now they all differ in the negative charge and the A and B acidic and amino sugars, right? So in all of them there is hyaluronic acid, which is actually does not have, number one, it is an exception that it does not have any sulfate group in it, which means there is no negative charge. Negative charge does not have any negative charge. And second thing, it does not attaches to the protein. It does not attaches to protein, which means in the extracellular matrix it flows freely like this. That is your hyaluronic acid, right? So hyaluronic acid, it actually present in where? It is present in your synovial fluids, and synovial fluid is present in your joints. So joints basically have your hyaluronic acid concentration, right? So when this concentration goes down, your joint starts to experience pain, right? Or this is also present in the eyes also. So this is the hyaluronic acid. So this is important about the hyaluronic acid. Sometimes question can be asked. It does not have sulfate group, due to which it does not have any negative charge. It does not attaches to the protein. Whereas other all type of the GAGs, they actually attaches to the protein, right? Next important one are your heparin and heparin. So heparin sulfate, this one is called heparin sulfate. So heparin sulfate is actually helpful in the wound healing. Wound healing, right? And this one is highly charged, highly negatively charged than other GAGs. So sometimes question can be asked, which one is the least charged and which one is the highly charged GAG. So heparin sulfate is the highly charged heteropolysaccharide, and it actually helpful in the wound healing. Then comes to the heparin. Heparin actually, it is present in the intracellular granules of the mast cells. Mast cells, right? And due to that, its function is the clot inhibition. Clot inhibition. So it actually inhibits the clot formation in the body. So these are the important heteropolysaccharides you should know about, and they are present in the body and very, very important. So number one is hyaluronic acid, which is present in the synovial fluids or in joints, right? It does not have any sulfate group, and it does not attaches to the protein. Next, heparin sulfate. It is helpful in the bone healing, and it is highly negatively charged. Next one is the heparin, that is actually present in the mast cells, and these mast cells actually helpful for the inhibition of the clot. Right? Next one is the keratin sulfate. It is not your keratin sulfate which is present in the hair. It is different than the keratin. Right? So keratin sulfate is different. Chondroitin sulfate, it is also different. So now let's see their structure. What are their structures? So your heteropolysaccharides, they actually have, number one, as I said, they have negatively charged. So hyaluronate, it has low negative charge. It is less negatively charged, right? Whereas dermatan sulfate, it has extra negatively charged molecules on it. Chondroitin sulfate, it also has negative charge. Keratin sulfate, it also has negative charge. Then your Corantin 6 sulfate and your heparin. So the confusing thing here is your heparin. Second one, heparin sulfate. And third thing is your hyaluronate. So always questions comes from this confusing topic. So you need to remember their function and their structural units also. So in case of heparin, it is iduronate sulfate and D-glucosamine sulfate. So this unit structure also you need to remember, right? So in this lecture, we discussed about different type of disaccharides, right? Their examples, their functions. Then we discussed about the polysaccharides. Polysaccharides. In polysaccharides, we discussed its category, which is hetero, and its homo. In homo, we discussed about the structural and storage one. Storage and structural one. Right? Structure, we discussed about cellulose and the chitin one. In the storage, we discussed about the starch, glycogen, and starch and glycogen only. Okay? And next we discussed about the heteropolysaccharides. Those are your GAGs, and in that we had seen according to the negative charge and according to their functions how many types are there. So the important one, I told you, the triple three: heparin, heparin sulfate and hyaluronic acid. So you should know about them. So that's all for the today. Thank you.